Yes, I think Graham's done a great job of introducing Nicole, so over to you, Nicole.
Right, hello everyone. So, as already stated, my talk is basically a continuation from Graham's and we'll continue on to Owen's. And basically, I'm going to be focusing on how we can really incorporate the network knowledge of the underlying biology in order to try to improve our capacity to make predictions with crop growth models. So there are a number of things to consider when we're looking at these networks. First of all, we are really trying to describe this network in a mathematical way. And first of all, we need to understand what are these main components of the system. Those components will become the nodes in our network. Then we need to understand what are the connections between these nodes, ideally what the direction is of the interaction, what type of interaction that is, as well as that effect. So often networks are going to be modeled in a number of different ways. So you can either model them through a series of ordinary differential equations, in which case they're going to require a very high level of parameterization, or you can then take a much more simpler route and try to use a Boolean network in order to simulate these things, but that's going to be more like some binary switches. So in terms of this spectrum of extremes, so from Boolean to ODEs, the approach that we are trying to take is going to be a little bit more on the simple end, but we're trying to incorporate a little bit some of these features that we might have in ODEs. Basically, the consequence is that we're going to have a very minimally parameterized model. Yes, sorry. Okay, so if we are looking at the more traditional ordinary differential equations, you can see that even though we have a very simple network, so there are very few different nodes that we are actually looking at, it's actually disguising quite a large amount of complexity under the hood. So this set of equations has 19 different parameters, which are each attempting on some level to represent something about the underlying biology and how it works. So the problem with this is that we often don't actually know what these parameter values are. That could either be due to lack of funding or lack of technology in terms of being capable of representing these parameters. And as a result, you might go through and do a massive parameter screen to try to find a combination of different parameters that are going to fit with the biological data. So this is a figure that you might remember from last year, produced by Brodie Lawson. And he used Bayesian statistics to demonstrate a number of different parameter combinations can still fit the biological data. Now, as you can see here with the this final figure on the left, you can see that depending on the parameter combinations that you produce, it is possible that if you move outside of the existing biological experimentation, that you could start making some very different types of predictions. Another thing to consider is this idea of sloppy parameters. I really love this name. So in this figure here, you can see a number of different dots, and these are representing combinations of parameters. And the ellipses in this diagram is representing a set of parameter combinations that are going to give you a very similar prediction. So depending on the axis that you're looking at, you can have certain axes which are sloppy, which means that you can have a lot of variation in terms of the parameter values and still achieve a high level of accuracy. And there are other ones that are going to be a bit more stiff. So they're going to be far more restrained in terms of what they could actually be. So you could be thinking that, given that this is the behavior, if you are starting to incorporate at least some of those parameter values, that you might be able to dramatically reduce the set of parameter combinations that could actually fit with the data. The problem with this potentially is that if you get even one of those parameters incorrect, you can start to actually have very, very incorrect predictions. So given this backdrop, what we decided to do is to try to take a different approach where we're just looking at the structure of the network itself and trying to see how much can we predict just from that underlying knowledge. And again, another figure from last year, you can see that given the very simple network that I showed a little bit before, we are able to get something about the behavior of the system correct. And as we increase the complexity of the network, we were even able to recover many of these nonlinear dynamics. So that was very promising. Now, there are a number of cons and positives in terms of using such an approach. The first con is that we are unable to make quantitative predictions using this approach, so we are trying to really strip out a lot of those coefficient values. And also another result of that is that we're not really trying to represent what the underlying biology is that's going on here. So instead, what we end up doing is we take a look at the system, we expose it to different perturbations, and we can say something about the direction in which that system will move in response to those perturbations. Some of the pros for taking such an approach is that it actually becomes very intuitive to automatically generate these models, and it's very easy to scale the model depending on our knowledge, and it really does reduce down the computational complexity to run simulations on these things. So in service of that, I built an R package called psoup, which takes a network diagram and will automatically generate a series of mathematical equations to represent that network, and then allow you to go ahead and simulate it. So what I like about this is that it reduces the complexity for the user, and I personally think it's going to be a very nice generalizable method that could be used in a number of different situations. So with respect to that automation of building a series of mathematical equations, so I have here a little schematic of a very, very simple diagram. We've got a series of nodes, we've got a modifier here, we have stimulants, inhibitors, and necessary stimulants, which I'm not really going to explain too much, but basically I have a set of predefined terms which are used to incorporate the stimulatory, inhibitory, and necessary stimulants. And here I have a little example of how we might translate this specific diagram into an equation based on these sets of rules. Now, the modifier that I have here, this is what I was talking about when I was saying that our method is minimally parameterized. So this is the access point for the user and is the only opportunity that the user has to modify how that network functions. So it doesn't do anything about the forms. What it does is it allows you to place a weighting on particular nodes in order to represent something about the underlying genetics of that system. Sorry. Yep, so here I have a network which was produced by Christos Masantis. He's a PhD student who started earlier this year. So on Wednesday, he's going to be giving a presentation about how he specifically made this diagram. So he's going to go through that entire process, his process for validating this network, as well as he's going to give us some thoughts about how we might connect this network to the crop growth models that we were talking about earlier today. So he's really designed this network specifically for sorghum with the idea of connecting that with APSIM. So there are a number of things that are necessary in order to connect a model produced by psoup to APSIM. So the first was to generate a C# script that could then act as a managerial script within APSIM. So this has been done, and Christos has been playing around with getting the interface for that to work. He's also going to be suggesting a couple of different ideas we've been having in terms of really linking the output of psoup into propensity to tiller, which is at least our first entry point into getting those things to collaborate with APSIM. Okay, so on top of that aim for having psoup function in this gene to phenotype modeling framework that the COE is backing, there are also a number of different ways in which psoup can be used. So one is with respect to hypothesis comparison and experimental design. And I'm also thinking that it could be very useful in terms of as a starting point to produce even more complex models, because the whole thing is quite transparent. So I'm hoping that this is going to be used in the future as a tool to even explore network evolution. And sorry, one more final thing is that we are using psoup for in terms of connecting with other existing models is the work done by Taylor Wass, and he's going to be presenting that in a poster later tonight. And basically, he's using psoup as a brain in order to direct our functional structural plant model in how to develop over time. So the benefit of FSPMs is that it allows us to represent over time the dynamically changing architecture of the plant. So it seems quite natural for us to be able to produce a link between these two things. Okay, thank you very much.
Thank you, Nicole. Are there any questions? Bruce.
Very nice talk. I like the fact you're taking something very complex and trying to make it a bit more concrete. Have you also thought about using kind of spectral graph theory to kind of examine how stable some of the perturbations might be?
No, I haven't. So basically the way that we've implemented the equations there, they do always tend to end up in a very stable position. I don't know if you would have noticed in the very kind of simple equation that I put up there, we're really averaging out a lot of these effects. But no, I haven't done any analysis like that, yeah.
Yeah, because that way the user can kind of check out when they put their parameters in without actually running it through to see what critical function, what parameters have high sensitivity, for example.
Okay.
And you could check that out with the eigenstructure pretty quickly.
All right.
Very nice talk.
Anybody else got a question? Nicole?
Okay, thank you.